Brilliant. So I'm introducing Naz. She's a Oxford PhD student in our lab. So she's doing her PhD in purebred. She's working on, you will see, sarbecovirus, but also Nipah viruses and the adaptive immunology of this virus in their host. And on top of that, she's also involved in the EDI and well-being committee in our institute, make it more inclusive for all of us. So that's great, and you can go.
Thanks, Julia. So I'm going to talk to you a bit about some of the work actually we've been doing for investigating the role of SARS-CoV-2 NTD in transmission and immunity. So as we know, over the course of the pandemic, different SARS-CoV-2 variants emerged, and they were classified based on their immune escape profile and their risk to public health. And those that were of the highest priority were the variants of concern. And in the UK, the Alpha, Delta, and Omicron variants caused waves of infection, with subsequent Omicron variants now emerging. So we know that what contributes to these variants emerging are mutations within the SARS-2 genome. And in our group, we focus primarily on the spike protein. So we're using this to look at viral entry and determine host range, but also effects on antigenicity. So what's probably most well known is the role of the receptor binding domain, which is present in S1. And we've shown we have had some data that shows that if you look at individuals who have been immunized with 2 doses of a COVID-19 vaccine compared to wild type, the Delta and Omicron variants show a reduction in neutralizing antibody responses. And this also holds true even after a 4th or a 5th dose, where some individuals have had a booster with an Omicron-based vaccine, but for some of the more recent variants, we see a reduction in titer. So it goes back to what Julian was saying yesterday, that we're able to sort of reduce infection, but not transmission. And for the majority of these, we've been able to attribute these changes to mutations within the receptor binding domain. So for beta, we can attribute this to the E484K mutation, and for delta to the L452R mutation. But what's less characterized is the role of the N-terminal domain in transmission and immune evasion. So to do this, to have a look at this, I received a UKRCN Impact Award, and I've had synthesized some constructs of the wild type, beta, delta, and Omicron variants, and also made their individual NTD mutations in a wild type background. And we've been looking at this in the context of 2 separate groups, either the individual amino acid mutations in NTD or the deletions, which are known as recurrent deleted regions. And these map to 2 distinct antigenic sites, which have been shown to be important for immune escape. So an example of this is if we have mutations in RDR1 and 3 with this monoclonal antibody 48A, we still see spike binding, antibody binding, but not for deletions in RDR2 and 4. So what we wanted to have a look at first is, do any of these mutations or deletions impact viral entry? So using lentiviral pseudotypes, which Julia just described, we had a look at this. So this is what the entry looks like for the different variants. And when we look at the NTD mutations, we can see that there are varying levels of entry, but none of them are heavily impacted. And then if we look at the deletions using BA1 as an example, it was quite different because it has mutations across three RDR regions. And either individually or in combination with other deletions within BA1, we still don't see any impact on viral entry, suggesting that the NTD mutation deletions aren't necessarily essential for viral entry. But what about for immune escape? So to look at this, we have some monoclonal antibodies that were given to us by Katie Dawes' lab at UCL. And looking first at at wild type, we can see that these monoclonals neutralize wild type quite well. But then when we add individual mutations of BA1 into a wild type background and look at this neutralization, we can see that a deletion at position 143 to 145 confers some immune escape. And this is true when this mutation is by itself or with other deletions, and shows the same phenotype when we use the full length spike. So what does then this look like for polyclonal sera? So using sera from immunized, from vaccinated individuals, if we look at these deletions built in a wild type background again, actually it's quite difficult to pull apart any differences of that these deletions are having. And this is probably not too surprising because most of the antibodies are directed against the receptor binding domain. So to try and circumvent this, we have established an assay to deplete RBD-specific antibodies. So we take recombinant SARS-2 RBD proteins that are his tagged, conjugate it to nickel beads, and then incubate this with our polyclonal sera. We can then use a magnet to pull out those RBD-specific antibodies, and then what we should be left with are non-RBD binding antibodies. So what that then looks like is the bar on the left, the pink, is what the neutralization looks like for when all the antibodies are present, and the purple is the residual non-RBD binding antibodies that are left once we pull apart those RBD binding antibodies. So if we now look at this polyclonal sera, actually the effect of the deletion in BA1, either by itself or in combination with the other deletions, is now more evident and similar to what we see for the monoclonal antibodies. So what does this look like for some of the other variants? So for beta, we see a similar story where the deletion present in the beta NTD seems to have an impact on immune escape. But actually, the spike as a whole doesn't seem to confer any immune escape. And when we look at the polyclonal sera, where we've depleted RBD-specific antibodies, again, we see an impact of the deletion, but it's the spike as a whole has similar levels of reduction in neutralization compared to wild type, suggesting maybe a compensatory role of the other NTD mutations or a role outside the N-terminal domain. And then for Delta, we see a different story again, where none of the mutations that are present in the N-terminal domain seem to confer any immune escape with the monoclonal antibodies we have. But the spike as a whole does have some immune escape. So we thought maybe, again, there's a role outside the N-terminal domain that's contributed to this. So we looked at the L452R mutation, which is present in the receptor binding domain. But even here, we don't see any evidence of immune escape. Then looking at our polyclonal sera, here there seems to be a slight role of the 156 to 157 deletion and possibly even the mutation at position 142. But you can see that none of them amount to the same amount of reduction that we're seeing with the full-length spike, maybe suggesting a combinatory role of the different mutations in NTD or a synergistic role of both the NTD and the RBD in giving this phenotype that we're seeing. So just to summarize, I've shown you that although the NTD spike protein so the mutations in the NTD spike don't necessarily play a role in entry, they do play a role in neutralization, with different monoclonals showing differences in neutralization against variants. And if we deplete RBD-specific antibodies, this also reveals some key residues. So going forward, I'd like to investigate the role of these NTD changes in the context of VOX-specific immunization. So if we deplete beta-specific RBD antibodies from beta-immunized animals, do we still see the same response? And then also because of this difference we've been seeing with Delta and Beta, if we generate chimeras of the VOX, can we sort of pull apart the interaction between RBD and NTD? So at the moment, what we have is this as a platform, but I've built them as cassettes where I can then chop and change the NTDs and RBDs. But I'd also like to look at what a full deletion of the NTD looks like, because as we know for some other coronaviruses, coronaviruses, for TGEV, the full NTD is required, but for PRCV, it's functional with an NTD deletion. So with that, I'd like to thank my group and our collaborators, and just to point out, I have a poster looking at SARS-CoV-2, beta coronavirus host range, if you'd like to come and have a chat with me at lunch. So thank you.
Okay, does anyone have any question? Okay, maybe then I can start. So I was wondering, this is a really big question, but I was wondering if based on this data and what you're getting, you could advise for any prevention or health policies that could be taken into account for vaccination or treatment of infected people.
So I think in the context of at least monoclonal antibodies as a therapeutic, if we are able to find ones that sort of cross-neutralize both, so well, I will say the ones that I showed you were directed to a certain region of the N-terminal domain. So the KT has ones that target other regions of the NTD. So if we could find ones that are broadly neutralizing in that context, I think that would be quite useful.
And so has the position been determined where this antibody bind on the NTD?
At the moment, they're just predictive. So they're based on competition ELISAs of known monoclonal antibodies.
Okay.
So it's not yet known what those sites are.
Okay. Anyone else? So I guess I will ask the last thing. Are you planning of building this mutation, for example, for Delta in a live virus and then test in infection with cell line, what the result would be?
Probably not, because I think that's probably like frowned upon. But I do know, for example, Andrew Davidson, he has some clinical samples of individuals where they have like these NTD deletions from some infection. So we could get viruses that are already present like those, yeah.
Okay, thank you.